speaker, which is Dr. Uli Sorge. We heard her in a session before. The title of her presentation right now is Does Boric Acid Preservation of Milk Samples Affect Differential Somatic Cell Count Analysis?
Thank you. Good morning. So, like my second talk is now about a little method where we were trying to figure out whether it works. So what is it all about? So we know that the somatic cell count is an indicator of good udder health or not. And so we use them on a regular basis through monthly DHI tests or the CMT test. And when we are looking, and it is really helpful, but when we are looking at the somatic cells, we have to realize that it's not just somatic cells, but there are actually different fractions present. So you have lymphocytes, macrophages, and polymorph nuclear neutrophils, so PMNs. And depending on whether a cow is healthy and has a healthy udder, the cells present differ. So in a healthy udder, we have mostly macrophages, obviously some lymphocytes. But once a cow has mastitis, the PMN fraction shows up and increases proportionally. And so one of the things that is a new diagnostic that we can use is, or we hope to use, is that the changes in the proportions of the macrophages and PMNs will help us better identifying cows with an active immune reaction, so with an active mastitis. And it would be really nice to use this as a diagnostic tool or additional diagnostic tool to classical, let's say, bacterial or microbiological detections. The old school was to actually stain the samples and look under the microscope and count manually, but now there are different technologies on the market that actually make this happen for you. And so it would be really nice to use those samples or those methods to further look into the SCC. Most methods are actually, or the one that I'm looking at, which is the FOSS methodology, FOSS 7, that uses bronopol samples. That's the normal conservation that you would have with DHI samples. And with those ones, it seems to work quite well. However, in Germany, we use for the quarter milk samples that we get to the laboratory, we use boric acid in a very light concentration. And it's, you know, when you're looking at it, you are wondering, okay, does it work with this kind of conservation as well? You know, because we have more boric acid than bronopol in the samples, do we have a dilution effect? Obviously, they don't show up, you know, you don't take the samples and immediately you have the samples in the laboratory. Instead, they send it by postal services or something, and the SCC might be a bit frail for the diagnosis of the different fractions. So do we have a destruction of cells? Plus it's also acid, so is there destruction of cells? And so the aim of this pilot study was really to figure out, does it work with bronopol? So number one, how does it hold up compared to boric acid? Does it hold up against bronopol, the gold standard? Does it matter because because the quarter milk samples are taken manually, you don't always have the exact amount of the ratio that it's supposed to be? You know, does that make a difference? And then also, again, you know, with the whole postal services, that can be sometimes a bit slow. Does it, you know, how is it with storage time? Does it affect the cells and the results that we find compared to the gold standard? We figured we need about 100 samples, and because we wanted to have the same samples, we decided to take about 250 milliliters or 240 milliliters of milk from cows, and so we split it up into 8 30 milliliter aliquots. So number one was our gold standard, which is the bronopol, which is about 100 microliter in one of those tubes, and then you fill it up with the milk. And then boric acid, it's about 1.2 milliliters for 10 milliliters of milk. That's what the books say. And so we said, okay, in order to get 30 milliliters of milk for the machine to actually run the samples through automatically, we needed to use those vials. So we needed to have 3.6 to 30. So, and this is kind of we used for the time. So this is the day of the sampling, day 1, 2, 3, 4, and day 5. And then obviously because we wanted to figure out, does the dilution have an effect on anything? And so we had it with twice the concentration of bronopol or 3 times the concentration of, sorry, not bronopol, boric acid to milk. So these are our 8 samples. And so while we were on farm, we milked into those containers with a lid, and then we moved them and then manually split them into the 8 vials. And we repeated the sampling twice. So number 1 was a 300-cow dairy with where my poor technicians were racing against time at milking. That it is CMT, and then manually stripped about 250 milliliters by hand from each cow. And then we repeated the same thing in a much smaller dairy where we didn't race against milking time, where we just had them, and between milking we locked them up in the headlocks, and then got the milk and mixed them and transferred them into the aliquots. So again, so we took the samples on day 1 and analyzed in the laboratory with the somatic 7 DC. We analyzed samples for SCC and DCC, and then the bronopol samples were also analyzed for various nutrients or metabolites in it, which apparently is not able to do with the boric acid samples. And then basically I compared the samples. When we are looking at the samples, or when we looked at the samples, there's an internal validation. So it basically says, do I recognize the cells, yes or no? And most of the cells, as you can see in the first trial, about 90%. In the second trial, we were much better, was a good separation. And the nice thing is, you know, very positive. The somatic cell count, just general numbers of somatic cell count, were very highly correlated between the gold standard of the bronopol on the one side and boric acid on the other side, also over time. So it was very nice to see this one. The ones with the double or the triple boric acid concentration were slightly lower in the somatic cell, as you might expect with the dilution effect. But still, there was still a high correlation. When we look at the DCC, which is the proportion of PMNs, so this is kind of what we found, 58 plus minus 21, and the range was really big, so between 0 and 90. Interquartile range was a bit narrower. And if we are looking at it, you know, just bronopol against the boric acid samples on day 1 through day 5, again, very good correlation, but over time it slightly decreased, and then also, as you can see here, the correlations clearly decreased in intensity if you're looking at the different dilutions of boric acid to milk, or milk and boric acid. And so if I'm looking now at the difference between the, so this is the delta, the difference between the DSCC that we find in the bronopol samples and the boric acid samples, and then compare it over the somatic cell count. One of the things is that we see we have a massive variation up here. Remember, the average was about 58. So we have a difference of between up to 70 points, and then it decreases over the somatic cell count. But again, the decrease starts at about 1 million cells per milliliter somatic cell count. And so I looked at this and I thought, am I overlooking something? What's going on? Why is this looking the way it is? And then I suddenly thought, oh, maybe, you know, up down here, maybe it's where the machine says, basically, I'm not going to work properly, so 50,000 cells, so okay, exclude them, and not much change, because about 50,000 is about here, and this one wasn't even a 50,000 also below 50,000 somatic cell count. So I was like, this is not it. Maybe it's something else. And then I looked at the fat content of our samples that we carefully mixed on farm and realized that we didn't mix it maybe properly either on farm or in the laboratory, which hopefully we didn't, but regardless, no normal cow has 0.5 or 12% fat in her milk. And so I said, okay, maybe I should narrow it and just look at the perfect samples, which are in a physiological fat content, just to make sure that we have a proper mixing. And this is again the before, and voila, we have a much better looking picture if we actually use properly sampled and handled samples. And so anything that I'm showing from now on is going to be with the good samples, so to speak. And so if we're looking at these graphs, and please bear with me, so this is always, the y-axis is always the Bronopol DSCC, and on the x-axis is always the DSCC of, in this case, day 1, 2, 3, 4, and here 5. And you can see we have a very good correlation. Here with the dilution, we see that we have a much bigger fanning, especially here with if you don't perfectly take the samples like a machine and instead have a little bit less in your quarter mix samples, for example. So it definitely has an effect on it. So overall, I would say if you have good samples, the overall correlation between Bronopol and boric acid was good, also over time. But you really have to make sure that the dilution or the mixture of boric acid and milk is properly, because both can influence the somatic cell count and also the DSCC. And my problem with it is that really with the low SCC, you have a much bigger diversion or variability in the results compared to our, let's call it, gold standard. And the good separation alone for the analysis of the samples, whether the quality is met for a good mix or for a good sample, might not be enough to establish the quality control for boric acid samples. So if you slept through my talk until now, so basically what I want you to take home is it's actually quite promising. It seems to be that the boric acid or samples conserved with boric acid might work, but you really have to make sure that your samples are really well collected, really well mixed, and you really need to find something that you can use if you use boric acid. And unfortunately, like I said, the machine doesn't allow for the metabolite analysis of the metabolites with boric acid. And so I'm not sure what the quality control will be, but just making sure that your samples work, and then you have good results. So with that, I say thank you very much.
Thank you very much, Ulrike. So are there any questions? If not, I have one, and sorry, it might sound ignorant, or maybe I have missed it. Why there is boric acid and why there is Bronopol? So
oh, yeah. So Bronopol is the standard conservative for milk samples that you take for the composite milk samples during monthly milk testing. Mm-hmm. And the boric acid seems to be the more it's the standard one that they use for microbial analysis in quarter milk samples in Germany. That's the one that they use. And yeah, they don't use Bronopol for it.
Okay, thank you. So, any other questions? If not, then I say thank you very much. And our next speaker is Masafumi Enokidani with his presentation, Analysis of Factors Affecting Milking Claw Vacuum Levels Using a Simulated Milking Device.
Hello everyone. Welcome to Sapporo, Japan, and enjoy to eat Japanese dairy products, especially sweet cake. So my presentation title is Analysis of Factors Affecting Milking Crop Vacuum Level Using a Simulated Milking Device. Background: NMC, National Mastitis Council, recommended to evaluate milking system performance with 4 steps. Step 1, observation test with machine learning and not learning. Step 2, dry test with machine learning but not milking. Main test factor is airflow in milk line and vacuum lines. Step 3, wet test with machine milking. Main test factor is airflow, milk flow, and milking technique. Step 4, flow simulator test for evaluating milking system performance and optimizing operating vacuum. Milking system technician must know basic factor of milking system. We will show the evidence of basic factor for observation test. What is the matter in this picture? I will clarify the important factors of milking system from now on. Material and method. We used flow simulator like this picture and milked with using bucket with flow meter and 4 simulated teeth. Flow simulator, simulated milking, vacuum measurement devices. We adjust at 30 seconds after starting simulated milking. Mean crop vacuum was measured at 2 minutes after wait this 30 seconds until stabilize the flow. Analyze factor and water flow rates. Water flow rates have 5 steps, 1.9 kilogram to 8.7 kilogram per minute. Analyze factors are operating vacuum, high line system, low line system, high line system with ATO, automatic takeoff, milk tube length, milk tube diameter, and aperture diameter, tubing configuration, vertical tubing, level tubing, milk tube lift height, flow type, and milk sampler setup. This slide shows basic factor of analysis and flow rates. Oh, sorry, mistake. Yeah. This slide shows factors of operating vacuum. Red line is on high line system curve, and black line is on high line curve with ATO. Both high line system crop vacuum starts at 50 kilopascal and go down around flow rate quickly in by lift vacuum loss. High line operating vacuum depend on lift vacuum loss. With ATO, high line system go down more than red line curve. Yellow line and blue line curve are on low line system. Crop vacuum do not go down with lift vacuum loss. Yellow line curve has with 60 millimeter diameter milk tube. Blue line curve has with 22 millimeter diameter milk tube. Then low line operating vacuum is lower than high line vacuum. Purple line curve is dual vacuum milking system on high line, named Superline system made by Orion Company in Japan. Milk line vacuum is 60 kilopascal, higher than common high line vacuum. Then vacuum lift loss is smaller than common high line system. Put on NMC recommended crop vacuum range on this graph. On highland system, low flow rate area means over milking times. And high flow rates area mean poor heat and massage time. We have to get operating vacuum in NMC recommended flow vacuum range on every milking system as possible. We have to use deciding operating vacuum with using flow simulator at installers and travel times. On this picture, row line system and dual vacuum system operating vacuum are too high vacuum. This is operating vacuum at 42. This slide shows the factor of milk tube lengths on high and low line system. On high line system, milk tube lengths have 248, 288, 328, and 317 centimeters with ATO. The longer milk tube make low flow vacuum more over with ATO. On low line system, tube length has 200, 250, 300 centimeters. The longer milk tube make low flow vacuum similar. Upper side picture shows two long milk tube lift, long milk tube, and aperture diameter. Lower side picture shows two short milk tube and poor flow alignment. Short milk tube length better than longer, but two short milk tube sometimes makes poor flow alignment like this picture. This slide shows the factor of milk tube diameter and narrow points on high and low line system. On high line system, milk tube diameter has 40, 60, 60 with milk checker. Small difference on aperture diameter occurred, big difference on high line system vacuum like a bottleneck. On low line system, milk tube diameter has 14, 16, 18, 22 millimeter. Smaller diameter make low flow vacuum clearly. Upper side picture shows mastitis checker. Lower side picture shows all type of vacuum cutting devices with narrow point. This slide shows factor of milk tube on low line system, of milk tubing on low line system. Level tubing is lower than vertical tubing. Upper side picture on vertical tubing shows that milk flow from up to down. Lower side picture on level tubing shows that milk flow from left side to right side. This slide shows factor of milk tube lift height on low line system with same milk tube diameter and length. Milk tube lift height have from 0 to 80 centimeters. Higher lift heights makes low flow vacuum. These pictures show milk tube lift height test on dairy farm. Upper side is 40, lower side is 16. This slide shows factors of claw type and sampler setup. Black line curve is top flow with sampler, and blue line curve is top flow without sampler. Difference between black and blue curve is sampler setup on top flow. Red line curve is bottom flow with sampler, and green line curve is bottom flow without sampler. Difference between red and green curve is sampler setup on bottom flow. Difference between black and red is claw type with sampler setup. And difference between blue and gray is claw type without sampler set. Bottom type flow vacuum is higher than top flow type in spite of sampler setter. Bottom type flow vacuum is better than top flow vacuum on low-line system. Conclusion. Combination of flow simulator and vacuum measurement device could analyze the factor affecting the milk flow vacuum level by changing flow rates. This method can apply for diagnose the performance of the milking system. Milk tube length should be as shorter as possible with optimal flow alignment. Milk tube diameter should be over 16 millimeter, especially over 18 millimeter for low-line system. Milk flow should not conflict with gravity. Bottom flow flow is better than top flow flow. Constricted parts like a mastitis checker, milk meter, vacuum cutting device should be carefully installed. Do you see any issue on this picture? A narrow